Welcome dear students. In this video we are going to learn about the second mechanism of G protein coupled receptor signaling, that is by rhodopsin. In my first class I have mentioned how muscarinic acetylcholine receptor pathway actually reduce the contraction rate of heart muscles. Now in this class I will talk about how rhodopsin work in the rod cells of our eyes. So in the rod cells there is a G protein coupled receptor present in a membrane. Let's assume this is a plasma membrane. This rhodopsin is actually the G protein coupled receptor. The rhodopsin protein can be divided into two components. One is called opsin. This opsin is the actually main G protein coupled receptor. This opsin is equals to actually G protein coupled receptor. Okay, and this rhodopsin contain another component that is called retinal. This is the light sensitive pigment retinal. It is actually modification of a vitamin A. This retinal is actually light sensitive pigment. Okay, so I can write here that rhodopsin is actually these two components, opsin and retinal. Now this retinal can be divided into two different states. One is called 11-cis retinal and another is called all trans retinal. By the way, this retinal pigment is actually attached with this opsin at the 296 position of amino acid lysine. 296 of lysine. You can remember it as a just information. Now this retinal can have two different state or structure, isomers. You can also see isomers. One is called 11-cis retinal and all trans retinal. What is the difference? Here is the difference in its structure, but you don't have to remember the structure. Just remember the fact that 11-cis retinal is present when the rhodopsin in dark state. So when we are dark conditions, the retinal is in 11-cis retinal condition or isomer, and when light falls, this isomer actually changes into all trans retinal. The fact is here, when retinal becomes 11-cis to all trans retinal, it occurs when light falls. So when rhodopsin receives photon, then this 11-cis retinal become the all trans retinal, and in that case this rhodopsin become activated. So we call R star, that means activated rhodopsins. So let's suppose this is the rhodopsin, as because we know this is 7 transmembrane protein, 1 2 3 4 5 6 1 2 3 4 5 6 7. Okay, so this is the N terminal and C terminal. This is the opsin protein, and here maybe some the retinal can be present, and when light falls, this retinal change from 11-cis retinal to all trans retinal, and this opsin protein, actually the GPCR, becomes activated. And when these become activated, it actually changes its structural actually conformations. When there is in darkness, then the G protein that helps in this transduction or signaling called G alpha T associate with beta and gamma, as in other cases. Like in my muscarinic acetylcholine receptor pathway, I have mentioned that G alpha I here is the G alpha protein, because this is the trimeric protein. Here alpha, beta, gamma, 3 subunits are present. This G alpha protein in case of rhodopsin called T, G alpha T, T for transducin. When there is no light, that means in darkness, this G alpha T is associated with a GDP molecule, because as it is, the name implies, G means guanine nucleotide. So it is associated with GDP, and I have mentioned in my basic class that when G protein associates with GDP, it is actually off in state. In that situation, it actually binds with the G beta. But when light falls, the structural changes in the opsin actually attract these G alpha T proteins to its close and then this close proximity, and then these G alpha T proteins attach with this opsin or GPCR. I can write here GPCR. Then the GDP is exchanged with GTP. That is called GEF, or means guanine nucleotide exchange factor. So this opsin acts as a GEF. So here also the same fact is true for muscarinic acetylcholine receptor. Here also this GPCR acts as a GEF, and it actually exchanges the GDP with the GTP. Now this becomes active. The theory says that when G alpha protein dissociated from this G beta gamma and actually attached with GTP, it becomes active. Okay, I am actually removing this beta gamma. This is not significant in this case, but in case of muscarinic acetylcholine receptor, beta gamma also plays a role because it activates the potassium channel. Now this G alpha T protein becomes activated and it activates a effector protein, because in my previous video I have mentioned that in case of G protein coupled receptor, the first thing is GPCR, then it actually attached with a ligand. So the receptor is GPCR, ligand bind, then it activates the G protein, that means G alpha, then G alpha activates effector protein, then effector protein activates the second messenger. Okay, second messenger, then second messenger activates some kinases or any other molecules. The same is true for here. When ligand, here ligand is the photon when falls on this rhodopsin, means opsin and retinal actually changes its conformations, then it actually activates the G alpha. Here in case of T, then it activates the effector. In this case the effector is called cGMP PDE. The full form of PDE is phosphodiesterase. PDE means phosphodiesterase. That means this molecule can break the cyclical nature of cGMP. That means this PDE can convert cGMP into, this is actually enzyme, they can convert cGMP into GMP simply, guanosine monophosphate. This is the signaling. Now how this PDE gets activated by this GTP? The molecular mechanism is here, that PDE, cGMP PDE means phosphodiesterase have actually 4 subunits. One is called alpha and it's called beta. This is actually active components, but it is actually associated with 2 inhibitors called gamma. This alpha is associated with 1 gamma, beta is associated with 1 gamma. Then these G alpha T proteins actually attract these gamma proteins. Another G alpha T proteins attract these gamma proteins and remove these 2 inhibitors from the alpha beta subunit. Then the PDE is only have this alpha and beta subunit, and then it is actually active in conditions, because this G alpha T, activated G alpha T, when associated GTP, it attracts this gamma or inhibitor. Then it actually reduces the GMP. So what is the significance of GMP in this signaling? Yes, there is a significance. In the membrane, plasma membrane of these rod cells, there is a non-selective channels are present. Non-selective channels, cationic channels. Through which sodium and calcium actually enter inside the cell. So sodium and calcium enter inside the cell, but this channel can be closed and open in conformations. When there is cGMP binds with this receptor or channels, at least 3 to 4 cGMP molecules should bind with this channel, then this channel actually becomes open in conformations. But as you can see, when light falls, this level of cGMP actually reduces. Then this cGMP cannot bind with this channel. Then this actually flow of the sodium and calcium actually stop. When sodium and calcium entering inside the cell stop, then the whole mechanism, actually rod cell is actually neurons that can secrete neurotransmitter stop. So when light falls, rod cells stop sending or secreting neurotransmitter. Brain actually interact or interpret this mechanism that when these rod cells are not secreting neurotransmitter, his brain sense that now it is light, there is light. When there is no light, let's suppose there is no photon is present, then this 11 cis retinal do not become all cis retinal. So this G alpha protein do not become activated. It is associated with GDP and with the beta and gamma subunit. Then this PDE is not activated. Then cGMP level is high. This cGMP can bind with this channel. Then sodium and calcium can enter, because when cGMP binds with the channel, this channel becomes open. Sodium and calcium can enter, and the cell of the rod cells actually have a potential action. The membrane potential become minus 30 millivolt. That is close to the depolarized state. As you know that each and every cells have a resting membrane potentials. The range is minus 60 millivolt to minus 90 millivolt in many cells, like in neurons and different cells. Approximately you can say that the resting membrane potential is minus 70 millivolt. Now you can see, in case of darkness, when there is no light falls in this opsin, then the channel is open and sodium enters, and the potential of the membrane becomes minus 30 MB, and that is actually quite high than the resting membrane potential. And in that situation, these rod cells can secrete neurotransmitter, and brain sense the neurotransmitter as a darkness. You have to understand this. When rod cells secrete neurotransmitter, brain interpret this as a darkness. When rod cells stop secreting this neurotransmitter, they sense that it is light. So this is the whole story about these rhodopsins. But how this mechanism stop? Because this is called visual adaptations. When light falls, then this mechanism should actually recover itself, because it has been seen that a single photon can activate this mechanism, and it actually takes only 50 milliseconds to become to complete this cycle. That means to become this 11 cis retinol to all trans retinol, and then it becomes this 11 cis retinol to all trans retinol to 11 cis retinol, because then it can also receive another photon. So this should be stopped. This mechanism should be stopped, and there is three way. I will show you here. There is three way by which this activation can be deactivated. The first is called gap protein. As I have mentioned that G alpha T protein, always the G alpha protein has intrinsic GT-page activity. That means it can act as a gap, but the rate of hydrolysis is slow. When other gap helps in this process, it actually accelerates the hydrolysis of GTP with GDP. You have to understand this. There is a gap present in the rod cell called RGS9, which associates with another protein called G beta 5. These two proteins associate as a gap, and it actually helps in the formation of or transformation of G alpha T GTP into G alpha T GTP in its inactive form. That is one mechanism. The second mechanism is when calcium levels or sodium levels falls, some proteins actually get activated. These are called guanylyl cyclase activating protein. When the level of calcium or sodium falls, this protein actually gets activated, and it activates the guanylyl cyclase. And when guanylyl cyclase gets activated, it converts GTP into cGMP, and then the cGMP can bind with this channel, and the channel can open and this can enter, so it can sense that it is in dark conditions. So this is called adaptations. Another way, third way, is that there is a kinase enzyme that is called rhodopsin kinase. This kinase enzyme can phosphorylate the C domain of this GPCR. You can see here the C domain can be phosphorylated at particular serine residue. Two serine residues are present at which the phosphate group can be added by this rhodopsin kinase. Okay, so then when this phosphorylation occurs, a protein called arrestin bind with this phosphate group arrestin. this arrestin when bind with this phosphate group, then G alpha T GTP cannot associate with this GPCR and cannot get activated. So this is all about the GPCR signaling in case of rod cells by rhodopsins. Hope this video will help you. Thank you for watching and always keep learning.